All right, let's talk about the muscular system. So, to overview the three types of muscle tissue you have in your body, you have cardiac, smooth, and skeletal. And we took a look at these all in our tissue unit. So, we reviewed the characteristics of cardiac muscle and the characteristics of smooth muscle. But when we're talking about the muscular system, what we're focusing in on is our skeletal muscles. And remember, your skeletal muscles are attached to your bones by tendons. They're multinucleated, which means they have more than one nucleus. They're striated, meaning they have a striped appearance. You can control them, making them voluntary, and they're going to be surrounded and bundled by connective tissue. So, the muscular system is going to include your skeletal muscles, but it also includes the connective tissue components that help keep those muscles in place. So, the functions of our muscular system are to produce movement, they help us maintain our posture, they support our soft tissues like our organs, they guard the entrances and exits to our body, they help us maintain our body temperature, and they're going to store nutrients. Um, so then we got into the anatomy of skeletal muscles, the different parts of our muscles. So it's important to remember again that our muscles are attached to bones by tendons. Tendons is just connective tissue that's holding them to the bones. And then we're going to take a look going from big, our entire muscle, to small, what makes up each part. So around the outside of our muscle, there's a connective tissue covering called the epimysium. And so this is just connective tissue that holds our muscle together and protects it from the outside. When we take a look inside of our muscle, the inside of our muscle is broken up into compartments by a connective tissue layer called the perimysium. The perimysium is going to contain blood vessels and nerves, and the little like bundles that form in the perimysium, those are going to be called your muscle fascicles. And each within each muscle fascicle, that's where we're going to find our muscle fibers or our muscle cells. Some other connective tissue components around each muscle fiber, there is again a connective tissue layer that's protecting it from its environment. The connective tissue that we find surrounding our muscle fibers is called the endomysium. And then around each individual muscle fiber, we're going to have the sarcolemma, or the cell membrane or the plasma membrane surrounding each muscle cell. So we just talked about our epimysium and our perimysium, and we talked about the endomysium. But an important thing to note on this slide, within the endomysium, we are going to find myosatellite cells, which are sometimes just called satellite cells. And what those cells are they're the stem cells that are a part of your muscular system that helps repair damaged muscle tissue. We also talked about myoblasts. Myoblasts are the embryonic cells that fuse together to form muscle cells. And that's why muscle cells are multinucleated, because of the fusion of several myoblasts together. You can see that in this diagram over here. All these black dots, these are your nuclei within one individual muscle cell or muscle fiber. Myoblasts that don't fuse together, they're going to become our stem cells or our satellite cells. So we already mentioned the sarcolemma. The sarcolemma is the plasma membrane around the muscle fiber. And then within the muscle fiber, we're going to find the sarcoplasm, which is the cytoplasm of the muscle cells. Around the muscle fibers, we're also going to find transverse tubules or T tubules. And what those are is they're just passageways through the muscle fiber. They're going to play a big role in getting calcium in and out of the cell. So this is just showing you the sarcolemma, the plasma membrane around each muscle fiber, and you can see the sarcoplasm, which is the cytoplasm. So inside the T tubules, you're going to find myofibrils. Myofibrils are the subunits that make up our muscle fibers. And there's two types of microfilaments found within each. We have your thick filaments, which are seen in this diagram, as the purple line, and you have your thin filaments. And we'll get a little more into those in a couple slides. But the myofibrils are what is eventually going to shorten to form muscle contraction. So again, we're still looking at our individual muscle fibers. And so, like I said, around each muscle fiber, we're going to have a sarcoplasmic reticulum, which is shown in the blue here. So this sarcoplasmic reticulum, its role is to store and release calcium ions, which is going to be needed for muscle contraction, and it's going to form a network around the myofibrils. It ends at the terminal cisterna, and when we put terminal cisterna together, we get a triad. So you can see that's a combination of two terminal cisterna. So if we go even smaller, we know our muscle cells are made out of myofibrils, but myofibrils are made out of units called sarcomeres. Sarcomeres are what contain your thick and thin filaments, and they're the smallest contractile unit of muscle cells. And this is what's going to be responsible for muscle contraction. So within a sarcomere, there's different regions of the sarcomere. We have the A band where you find thick filaments. The thin filaments are going to overlap a little bit in that A band. Within the A band, there's the M line that's going to hold our thick filaments in place. You have the H band. The H band is going to be the region of the A band where only thick filaments are found. And then you have the zone of overlap, which is where the thick filaments and the thin filaments are together within the A band. I have a picture coming up in a few slides that'll help us explain it. Then also within each sarcomere, there's going to be two I bands. I bands extend from one A band of a sarcomere to the A band of a next. And within the I bands, we're going to find Z lines, and Z lines are the boundaries between sarcomeres. It's important to note that Z lines are also made of actin, just like our thin filaments. And so here's a sarcomere similar to the ones that we drew in class. So like I was saying, you have your A band, which is the big region in the middle. Within the A band, you're going to have the H band, where only thick filaments are found. You're going to have the zone of overlap, where you have your thick and your thin filaments together. You have your end lines that are just holding your myosin filaments in place. We have our I bands with our Z discs or Z lines. And then off of those Z lines, we have our actin, our thin filaments coming in. So this whole unit, IAI, is called a sarcomere. And if I were to line a bunch of sarcomeres up, it would be called a myofibril. Here's another diagram of that. I really like this picture because what it's doing is going from big to small. So what we just talked about is we were looking at sarcomeres, the smallest contractile unit of muscle. And our sarcomeres are made of our thick filaments, which are made of myosin, and our thin filaments, which are made of actin. So when we put more than one sarcomere together, we get a myofibril. Several myofibrils are going to make up a muscle fiber. Muscle fibers are packed together in little compartments called a muscle fascicle. And remember, those fascicles are surrounded by the perimysium. And when we put several muscle fascicles together, that's making up our actual muscle. And remember, our muscle is surrounded by connective tissue called the epimysium. So taking a look at our thick and thin filaments, our thin filaments we know are made of actin, and there's several other protein components that are going to help form the actin molecule. So it's important to remember that on actin, there's going to be active sites, and those active sites are so the myosin can bind to the actin to cause muscle contraction. Normally at rest, your binding sites on the actin is covered by the troponin-tropomyosin complex. So what happens is the tropomyosin is going to cover the active sites on the actin molecule, and binding can only occur when there's a position change, when the troponin-tropomyosin complex gets out of the way. I like to think of them like as the bouncers. They're not letting anyone near actin until they get out of the way. So then our myosin is what's going to bind to the actin. The myosin is going to have heads that come off the molecule, and those heads are what wants to bind with the actin to cause contraction. And when they bind, we call their interaction cross bridges. So in this picture, you can see your pink, that is your myosin, and these little things coming off it, those are the myosin heads. And those myosin heads want to attach with the actin, and when they do, they're going to pull the actin towards the center of the sarcomere. So we call when a muscle contracts the sliding filament theory, and what happens to the different regions of our sarcomere when our muscle contracts is the H and the I bands get smaller. The zone of overlap, where their thick and thin filaments are together, gets larger. The Z lines move closer together. It's kind of like when you crush an accordion in to make sound. But the width of the A band is always going to stay constant. And the sliding filament theory states that thin filaments are sliding towards the center of each sarcomere along thick filaments. With this, it's important to remember that the myosin is grabbing onto the actin. The thick filament is grabbing the thin filament and pulling it towards the center. So in order for this to happen, in order for muscle contraction to happen, our muscle cells have to be stimulated by a neuron. And so what we call a muscle cell and a neuron together is a motor unit. Our neurons release neurotransmitters, which are chemical messengers. And those chemical messengers get released into the synaptic cleft, or the space between the neuron and the muscle cell. And the muscle cell is going to pick those neurotransmitters up. And those neurotransmitters are what is eventually going to cause contraction. The neurotransmitter that's used for muscle contraction is called acetylcholine, which we abbreviate ACH. Eventually, our muscle cells will break acetylcholine down so our muscles don't stay contracted. So where our neurons meet our muscle cells, we call that a neuromuscular junction. And remember, our nervous system, our brain has to tell our muscles to contract. So we call the interaction between our neurons and our muscle cells a neuromuscular junction. At the end of our neurons, there's going to be synaptic terminals. Synaptic terminals are filled with acetylcholine, our neurotransmitter, and can be released from the synaptic terminals. The space between our neuron and the muscle cell is a synaptic cleft. So acetylcholine gets released from the synaptic terminal, travels across the synaptic cleft into the muscle cell. On their actual muscle cells, there's something called a motor end plate. And those motor end plates have receptors for acetylcholine. And once those receptors have acetylcholine, we can start the process of muscle contraction. There is an enzyme within our muscle cells called acetylcholinesterase, which is going to break down acetylcholine in the muscle cells so we don't have that continued contraction. So here's how all of that works. So step one, an action potential, a signal from our brain, arrives at the synaptic terminal and our neurotransmitter acetylcholine gets released. That acetylcholine is going to diffuse across the synaptic cleft. It goes from the neuron into the muscle cell. The acetylcholine attaches to the receptors in the motor end plate of the muscle cell. The action potential is going to travel into the T-tubules of the muscle cell, which are our passageways, causing the sarcoplasmic reticulum to release calcium ions. Once the calcium is released, acetylcholine gets broken down by acetylcholinesterase, which causes the muscle to return to its normal state. So we know how the calcium gets in and how that causes contraction, but now let's take a look at our actual muscle cells and how they contract. I can skip over this. So when the calcium ions enter the sarcoplasm or the cytoplasm, they bind to troponin, causing it to change position and allowing the interaction between the actin and myosin heads. Because if you remember, the active sites on actin were covered by the troponin-tropomyosin complex, and this calcium is going to move it out of the way, which allows myosin to bind to actin, creating our cross bridges. And then the myosin heads are going to pull away from their M line, and the myosin head gets to like a cocked position. It's ready to go, which requires energy or ATP. When it gets released from that position, it's going to attach to the actin and it creates something which they call a power stroke, which pulls the actin filaments towards the center. Then the link between the actin and myosin is broken. The myosin is going to reactivate, so this can happen over and over and over again. Here's a diagram showing that action. So for our muscles to relax, acetylcholine is broken down by acetylcholinesterase. The sarcoplasmic reticulum is going to absorb the calcium ions that were previously released. The calcium ion levels go back to normal, and the troponin tropomyosin complex returns to its normal position. So it's again covering the active sites on the actin molecule, and then contraction ends. So then we went in to talk about how our muscle fibers are at rest and when they're stimulated. So our muscle's tension is defined by two things: the muscle fiber's resting length and how frequently it's stimulated. So different muscles have different tensions, and we can look at their twitch graphs, which is just like we call a twitch a contraction relaxation cycle. And there's different types of muscle tensions that we discuss. You have a TREP, a wave summation, complete tetanus, and incomplete tetanus. We took a look at tetanus when all the muscles in your body are constantly contracted. And we also discussed rigor mortis, which occurs after death when there's not enough ATP to detach the cross bridges to take apart the myosin from the actin, so your muscles get locked into place and you become really, really stiff. We then took a look at different types of contractions. There's two types of muscle contractions. There's isotonic contraction, which means when the muscle contracts, its length is changing. So when you lift weights, you're changing the length of your muscle. Your muscle shortens as it contracts. And then there's isometric contractions, where you're applying tension to your muscles, but you're not changing the length of your muscles. So some examples of this are if you're carrying heavy objects, just standing up, your muscles are contracting, but you're not changing their lengths. Then we talked about ATP and how we need ATP for our muscles to contract and where we get that from. We looked at aerobic respiration, which is the best way to get ATP for our bodies. It creates the most, but we have to have oxygen for it to occur. If we don't have oxygen or we have an oxygen debt, we're going to have to go into anaerobic respiration. It produces less ATP, but it still gives us some. But we do get lactic acid as a byproduct, which makes our muscles sore. We took a look at muscle fatigue and what happens when your muscles get tired. The different types of conditioning you can do to strengthen your muscles. We looked at the two different types of muscle fibers you have. You have fast fibers, which are most of your skeletal muscles. They're going to reach their peak tension really quickly. They have powerful contractions, but they get tired really easily. You also have slow fibers, which take a little longer to contract. They're smaller, and they contract for very long periods of time. We looked at two pathologies you can have with your muscles: hypertrophy, where your muscles become very enlarged, and atrophy, where there's a reduction in size. And then we looked at muscle terminology. If we were going to learn about all the different muscles you have in your body, you'd have to become very familiar with this terminology. You need to know that an origin is where a muscle is attached to the bone. I like to think about the origin is when you move your muscle, that's the bone that stays still. And then the movable location where your muscle is attached to bone is called the insertion. And then muscles work together or they work against each other. So the muscle that you're moving is called the agonist. The antagonist is the muscle that's opposite of the agonist. So I can think about it if my bicep is the agonist, my tricep is the antagonist. It's doing the opposite of the muscle I want to move. And then your agonist is going to be assisted by other muscles called the synergist. And that's all you need to know. Watch this video a couple times, make your note sheet. Your note sheet can be an 8x11 piece of paper, double-sided. Look over the diagrams that we drew. Watch the Muscular System Part 1 crash course on YouTube. It does a great explanation. and let me know if you have any questions.